Welcome to Clay and Conversations at the Clay Studio. I am Jennifer Zwilling, the curator and director of artistic programs, and I'm very excited to have Marguerita Hagan and Carsten Skarke. I didn't ask you how to pronounce your name.
Skarke.
Skarke. Is that better? Okay. Thank you. We are very excited to talk about your project. First, I'm going to let everyone know that we recognize that the Clay Studio stands on the indigenous territory known as Lenapehoking, the traditional homeland of the Lenni Lenape people. We would like to take a moment to reflect on the role of the Lenape as the past, present, and future stewards of the land, and the role of all of us to join in that tradition of respect and caring for the land and each other. We hope that everyone will come and visit our current exhibition, Clay as Care. Speaking of caring for the land and each other, we are having a symposium on Saturday, October 25th, that will focus on that exhibition and the artists who have been working for the last 3 years on their pieces for that show. And that show is why we, at this moment, decided to invite you two to come and talk about your important project, which I'm just going to keep teasing people about because we're still not ready to start talking about that. Hi. Okay, so I'm going to introduce our guests. Marguerita Hagan is a ceramic sculptor based in Philadelphia. She's an activist for the thriving of all life in mutually sustainable communities and environments. The concept of interdependence plays throughout her sculpture, teaching, and community arts. Throughout her career, Hagan has brought to light the beauty and engineering of our planet's diverse ecosystems and our powerful role as stewards. At this climactic time of change, her work focuses on the largest and essential element on Earth, the ocean, from micro to macro. The intricate ceramic forms shine light on the wonder and respect for the fragile, diverse life with which our lives are intrinsically linked. Hagan's practice is an ongoing discovery, magnifying our awareness, reciprocal responsibility, and protection of each other and our planet. She received her MFA from the Pennsylvania Academy of the Fine Arts and her BFA in ceramics at James Madison University. Her projects include collaborations with scientists, artists, and community, environmental art science residencies and lectures. Her work is in private and public collections and exhibited nationally and internationally. Welcome, Marguerita.
Thank you.
Carsten Skarke is the Robert L. McNeil Jr. Fellow in Translational Medicine and Therapeutics and currently an adjunct associate professor in the Department of Medicine at the Perelman School of Medicine, University of Pennsylvania. Carsten's research focuses on the role of biorhythms in health and disease and how these are perturbed by environmental factors such as air pollution. His mechanistic approach spans the full spectrum of time-specific multi-omic strategies integrated with multimodal phenotypic data to parse the human chronobiome. I can't wait to hear more about that. Lessons learned from establishing these comprehensive clinical study paradigms in adults will be applied to children in the coming years. I also want to note that Marguerita is hosting Post this weekend at her studio. You want to tell us more about that?
Yeah, open studio tours West Abroad is noon to 6 on Saturday and Sunday. Everybody is welcome. And I'm also doing a reception from 5 to 8 Saturday. And during the reception at 6:30, Karsten and other scientists and I are going to be sharing the shields that we'll have out at the house and the studio. And Sunday from 2 to 3:30, I'm hosting an event with the director of the American Swedish Historical Museum and CMAC director. They're doing a collaboration, immigrants helping immigrants, to for an exhibit that will be at the Swedish Museum in January of art made by immigrants. Yeah.
Great. Yeah. Thank you. So I'm going to start with a version of the same question that I ask all of our guests, and that is usually how and why did you make the brave decision to make your life in art? But we're going to ask Carsten to start and to shift it a little to science.
Thank you. Yeah, thank you for the opportunity to talk about our work today here. Welcome, everybody. So to your question, that starts a long time ago. I have to go back to high school in Germany. where I didn't have a good sense of what I wanted to do professionally. And so I had back then a chance to do a training as a paramedic, and that exposed me to the world of medicine. And I really fell in love with medicine. It was like the practical side of it, the interaction with humans, and then extending that into understanding the diseases, understanding treatments, etc. So I started med school. And during med school, I always knew I wanted to become a practicing physician. But then I started to work on an MD thesis and did research. And that way, I learned how I loved doing research. And then I did a couple of years practicing as a physician, but then I had a chance to go into research to check it out if I really like it. And I stayed in research ever since. And so a theme there is really this interactive approach, this hands-on approach, which really opened my eyes for me what I like and what I didn't like. And so that informed also our work and the things how I approach in nowadays.
Yeah, that's great. Thank you. I mean, I'll just ask one more sort of follow up, which is, we talked a few minutes ago about the connection between science and art and the creativity that is needed when you're a scientist who's thinking of a research topic or problem solving. Or can you talk a little bit about that those early years, maybe as a kid, or when you first started in science, and your sense of a desire to be creative, or that you are a creative person? Was that something that you felt was part of your persona?
Yeah, so that's a wonderful question. And the way I would respond to that is that looking back, I always felt the urge to, once I've done something for some time and I thought, that's a job well done, I felt this urge to make it, or to break out of the norm, the normative, and to approach that from a different perspective, to like reinvent it, but in a new way. Just for, yeah, I mean, out of curiosity to see if there are ways to improve an already well-working system. That's like on a higher level. And then on a day-to-day level, I love to dance, social dances, salsa, swing dances. And then when it comes to research, that needs quite some high level of creativity, because I mean, that's my own observation, that over the past decades, the research outputs, the way we do research has increasingly become more complicated and more comprehensive. I see that in my own work, where when I started, I was handling a small number of data points, right? Hundreds, maybe thousands, right? And now we're like in the hundreds of millions, right? And so that is a level where it just gets very complicated. And so the question is, how do you show your science, your work, in a way that not only your colleague can understand it, who's working with you, but the general public, right?
That's great. And we're going to pick back up on that last comment after we hear from Marguerita and when we talk about the project you're doing together. So Marguerita, how and why did you make the brave decision to make your life in art?
Well, I feel like it found me, but I grew up in a household with art was really revered and a part of everyday life. And my mother is an artist. Her mother, my grandmother, Lena Burke McNamara, was an artist and born in 1890 and went to the Corcoran School of Art, and then went to PAFA and studied with Cecilia Beau. But for me, I didn't always as a child really feel like I was really an artist because my sister was the one drawing and painting with my grandmother, and it really wasn't my calling. But as when I was probably 6, I remember finding clay on the beach behind our house. I grew up on the Lafayette River in Norfolk, Virginia, and I made little pots and made shells with the driftwood in the bushes along the river. And I loved going to Little Beach. Then in high school, there wasn't any clay being offered, but I went up and probably was 13 or 14 years old, and I went up to a woman at a craft show out in a park and asked her if she would teach me how to do clay. And she said, I don't teach, but that woman over there does. So I went over to her booth and ended up riding my bike to her studio through high school. And Betty Kite is where I got my start. And then when I went to James Madison University, I knew the one class I needed to sign up for was ceramics. And that's when I met Misako Miyata, which was really an incredible investment in my life and opened up my doors. And I saw that this was the way for me to breathe and to live my life. So I just kept finding a way to keep sustaining it.
That's lovely. And since I pressed Karsten on the idea of his science having creativity, your creativity and art has always had science. So can you talk a little bit about your did you always know too that that would be part of your creative journey, that you were interested in those ideas?
No, but I love life and I love nature. Nature's always been a huge inspiration and a great passion to share the connection that we have with all life, from microorganisms to macro, and with each other. Um, it really was early on that when I snorkeling for the first time in tropical waters in Hawaii, I was like 25, and I was so moved by the sea life and the reef itself. And I was living on Whidbey Island in Washington State at the time, so I couldn't wait to get back to the studio and put these patterns on slip cast dinner plates. But then they evolved into a thing I called the shield, that I evolved wanting to capture a pufferfish, which is blown up. And so I found these like old industrial walks in a scrap yard, and it felt like the perfect form to do the pufferfish. And then I realized it was like a shield, like a tribal shield and an honoring. It's also a thing of protection. And I felt like this was a beautiful form to honor the species we share the planet with. So when I began Lemaire, I was going to do a group of these shields for Angela McQuillan, who was a curator at Astor Klein Gallery. And I like diversity. And to represent ecosystems around the globe. But 90% of these things that I was coming up with were from the ocean. And I was looking through my Ernst Haeckel book, Art Forms in Nature, and I opened up to this page that I was like, oh my God, these are shields. They're waiting to be made. And I had no idea what they were. But then I had to look into it. And so I do love research too so much. Then I found out they were one cell organisms that lived in the ocean, and they help provide every other breath we breathe. So I was just so crazy passionate to start this series. I named it Lemaire. Angela said, yeah, just go with it. So that was the beginning of Lemaire and the end of 2015. So since then, I have been just having a blast learning by the research, and we protect what we love. So the point is to share and magnify this unseen world that makes our life possible, so we'll take better care of it. Yeah.
Yeah. Well, you both ended with the perfect segues into talking about the project that you have been doing together. So the idea of visualizing your work so that not only your partner can understand it, but someone else can too. And the reason why the shield is the form that it has taken, and that you've shared that idea so that other people can communicate through the same form, which is lovely. So I want to talk about how your project got started. Do you want to start with the slides to talk about that, or do you want to preview the slides with a little bit of how did you meet and how did you conceive of this project together?
I mean, the slideshow contains
We'll have
that.
that. Okay, so we can start.
Maybe we run through that and then,
Okay.
yeah.
That sounds great. And I just also want to say thank you to Trinity Dubois, exhibition coordinator, who and Maddie, who has put together all of our tech today, and Trinity helped with gathering information. So thanks to you both. Okay, so you all can just say next slide when you're ready to go.
Wonderful, I do. Yeah, thank you. So at my work, I wear a hat as a scientist, but then also as an educator. And there I think about educating the next generation of scientists. And so, of course, I also think about the future as scientists. How will the profession be shaped in the coming years and decades? And so on the next slide, I direct a, So along those lines, right, about the future, the Pew Research Foundation, they each year do a survey among the US population, around 10,000, to ask general questions about how science and scientists are perceived in the public domain, right? And so just here highlighting the yellow-greenish bars on the lower side, that over the course from 2019 to 2023, the public trust is decreasing. And on the next slide, then looking at specific domains, and these two graphs on the left compare data from 2019 to 2024 on the right. And if you hone in just on the bar of good communicators, you will see that on the left in 2019, 54% of that sample thought that scientists are good communicators. And in 2024, that decreased to 45%, right? And this is a concerning trend, right? And so on the next slide, thinking about ways to address that, I thought about 4.5 to 5 years ago that we and I actually have a platform to approach this experimentally, I will say, as a scientist, right? So this is just a program for workforce development where we select competitively undergraduate students from various partner institutions, and then they spend 10 weeks with us with a mentored research experience in labs across the University of Pennsylvania and the Children's Hospital, right? And we supplement their hands-on research experience with a seminar series, with a professional workshop. And so, and this is just the picture of us, of the current cohort, or from earlier this year. And so then on the next slide, we came up with that thought or approach. How about we embed an artist in residency program with our undergraduate budding scientists, right? And we started that over 4 years ago. And then Marguerita was our artist in residence last year and this year. An exception, the 2-year stint. Usually we open up each year for new calls. But to cut a long story short, basically with Julie Rainbow, our first artist, then Angela McQuillan, our second-year artist, and then with Marguerita, were able to develop a successful program, and we'll show you the mechanics of why we think this is successful. On the next slide, and this is now Marguerita will take over to guide you through how this shaped up over the past 2 years.
Okay. We're seeing shields, ceramic shields that the current interns made for the 2025. We can go to the next slide. This shows the page from Art Forms in Nature in the center, Ernst Haeckel's microorganisms. These are drawings that he did. He was a 19th century philosopher, scientist, and artist that is really well known for his work. This inspired, you can see the shields just to the right of the image and lower right that were inspired by that, a page from my sketchbook. An actual microscopic image on the top right of a diatom, Odentella aurita, and my shield that I did below it. A couple of images from the exhibit at the Philadelphia International Airport, and the shields above. These were, I started with fauna, so patterns from creatures, and it has now grown into flora and fungi and lemare. And not only like a thing of protection, but the shield is also like a family crest as well, honoring lineage and a lens of an eye to focus on also our relationship as stewards. Next slide, please. I'm in an exhibit right now in Lausanne, Switzerland, a collaboration. I'm so really happy to work with Kathy High. She's a real art side pioneer. This exhibit is on microbiomes in the gut. And so Kathy had an incredible project where she was studying microbiomes and if they're shared within households. And these are 3 households that she studied. And these are the shared microbiomes, not only in the human, adult, infants, but also the pets. So I said I could put that into 3D for you, Kathy. So that's what we're seeing. And they've extended the show. It's up for almost like 2 years now. It's up through August of 2026. Next slide. These are just some examples of how the shield is played out and communicating different ways. This is me making the shields do different sizes. And for this project, I did 3 different sizes. So the interns and the mentors would have some comfort to choose a size that felt right for maybe their designer, also how much, the focus they wanted to put into it. Next slide. I'm using low fire clay for this and get the piece dried, and I set it up in the wok, and these pieces come out of the kiln ready to hang. I just put a coated wire, they hang very easily, and there they are going through drying, and go through an 04 bisque at 1900, like 45 degrees. Next slide. Then they'll be ready, because when I proposed this, the artist was shadowing the scientist in the project, in the residency, but I knew that they wanted to make this sustainable. And I felt like with these young scientists and doctors, the greatest way to make it sustainable, to have a greater reach to the community and building well-being, is to plant seeds with these young doctors and scientists. So I felt like if they could learn to talk about their science in a way outside the lab, it would be great, more successful, and the healing would reach more people. So I proposed to have the shields and have these young scientists that were paired with the mentors at Penn to their science in the ceramic with the shield. And they finally agreed to let me do it. I took the studio to Penn and had an incredible team of support with Georgina Senior, Sophia Sierra, and Colleen Itani this year. And here we are. And you can see I had the sample so they could see what things would look like in the finished state. There's so much science in ceramics, and that's it's a really great pairing. Next slide. The students, we Zoomed so they could do their drawings and prepare before they came. And so they came with drawings, and then they could draw in pencil, and we used underglazes so they could really paint their work on. And then they did a clear glaze on top. The time efficiency was very important because they have a very intense schedule, and I was really pushing them to let me do this. And I said, well, if I bisque fire them, you can do it in one session to do the underglaze and the clear glaze. And Carsten agreed. And we had they had a takeout dinner there, and we had music, and the kid, they really loved it. And a lot of them said, I was so stressed about all the things, but this is the best thing that's happened all week, or this is so much fun. So it really is a healing and a very much fun for everybody, I find, in my experience. Their drawings were great. We can go to the next slide. This is them just going through the process. And I had two wonderful supports with Ronan and Nadim that also work with Karsten. They work together on a shield. Next slide. And this is Danae from this year, painting and drawing, and then the use fans to help dry the underglaze for the next stage. Next slide. And they're painting. There we go, same process. Next slide. Yeah, next slide. So some of the students couldn't make it to that evening scheduled. So I had them come to my studio, and this was a really intimate and wonderful way to be with the students. And I had some young artists in the class that night in my own studio class room. So this was wonderful. That's Rebecca and Christina working on their shields. Next slide. And then when after the Penn studio night, a number of the students needed more time. So I said, okay, come on over. So they came over to the next week. I had an opening for them, and they did finish there. And Danae has the apron on the lower right, but she really wanted to come, and her shield was finished, but she was such a great help. She really helped us. We all worked together getting all the shields glazed and everything. It was a natural art and science collaboration that night with the class, the studio class, and the scientists visiting. Next slide. There they are, glazed, going into the kiln, and this is going to be an 06 glaze firing. Next slide. And there's a before and after. It's so much fun. I love kiln shots. Next slide. And then I brought the work back. Previous slide, please. Brought the work back to Penn, and they painted the lusters on. Now, lusters are magical. They go over the glaze fired work. And it is the glass and the silica and the glaze that makes a luster the magic that it is. Next slide. And these are shots on the left of the luster painted on, and on the right is the finished shield in the kiln, and that's a temperature. I did it at 019, which is a happy place between the 020 of the mother of pearl and the 018 of the gold luster and the silver. Next slide. So much science. We get physics and chemistry and engineering and electrical, and physics is so good. So these are the beautiful slides of the first year of 2024. And then the next slide will show us the shields from 2025. What I think we were all so blown away by was their presentations when they presented. And also want you to know that none of these students had ever done any clay, and many of them know art before. Next slide. So I was so determined and so excited to get some mentors also into the studio. Now, because they have such a greater depth of experience in their science and they are the guides for the interns, I wanted them to work in wet clay, which has a greater dimension. And so they got to carve and add. And this is Carsten in the center. And Nick Balderston, who was a mentor to Sneha, their focus of research is transcranial magnetic stimulation, TMS. And then on the right is Karen Pluquet. Next slide. Karsten's very involved piece, which he's going to spend some time sharing with you, but it was so fun to get Karsten in there. It was such an innovative piece, Karsten. So exciting. His first ceramic piece. Next slide. And here we are working this year. I've had more mentors, and I had 2 women scientists and 2 women from the ITMAT team. And Dr. Adrienne Caldeo is in the foreground with her 2 beautiful daughters who also did some work. And Maidani is over there in the upper right. She studies wastewater in CHOP. She's the only person doing the kind of research she's doing in the country. And Jessica down in the center with Karsten, who has been an incredible guide for the program. And then Colleen Itani on the lower left, who came on this year to work with the artists and science residents. He has really been an incredible gift to the program. Next slide. Maidani's work is just incredible. And again, no one had ever done clay before. Her research is up there. We'll see a bit more. And Jason Batavia came up. He was at the students' presentation of their shields at the ICA this year on July 24th. And he came up at the end and said, "Can I do a shield?" And I'm like, okay. So I worked him into another way to get in the studio, and he's a Dutch descent, and so his favorite artist is Van Gogh, and here he did. His work was inspired by Starry Night with the communication between the brain and the bladder. Next slide. This is the greenware or the wet clay drawing and the bisque of these particular shields. Next slide. And here we are working, and they're beginning to paint the underglazes on their shields. Next slide. And especially love these shields with Idienko's. You can see her image. She's a holistic dentist, and she's also a radiologist. So her look at the dental health is throughout looking at the whole system and the whole body. Dentists often are the ones that might find a tumor and really save people's lives. And then Jason's work is at CHOP with children and dealing with a lot more than I can share right now, but just really communication between the brain and the bladder and a lot more. But his boys came with him. So we really had a great community time with all of these studio sessions. Next slide. Here are the finished shields of the mentors. Yeah, so beautiful. Next slide. And here from this year. Next slide. Carson's going to share his own personal.
I'm up, yes. Yeah, so just to emphasize how well Marguerita managed to integrate her work with the students. I mean, when we started with that program, we did it in the typical science way, where we run small pilot studies, right? And so over the years, we got to understand better and better what the possibilities are of fusing art and science, in science and art, right? And Marguerita, the last and this year has been really a tremendous success in really sharing her experience and work with the students. And one way to think about that is that I look at this and I say, by doing this, we extend the vocabulary of our students to communicate their science to the public by incorporating the experience of adopting this clay work, and then also having this clay piece in their hand, and to um, put this in front of them to attract interest from the public, to engage the public through the art, and then further and further go into the actual science of what that signifies, right? That's, I think, a much more holistic or much more effective way to cast a broad net into the public in terms of generating interest to engage with the scientists.
Can I interrupt for 1 second? And you basically answered a question that was formulating in my mind, but I guess I want to underline the fact that you developed this program. You founded the idea of bringing artists in to talk to your students. Can you, and you've already answered it to a certain extent. You showed us the graph about the research showing that people are perceiving scientists and doctors as being less able to communicate. But do you remember, or can you talk about that moment of, aha, this is what I want to do? Oh, we can solve this problem, or not solve it, but address this problem by bringing artists in. Did you see it happening somewhere else? Was it just something that came to you? I'd love to hear the generative moment.
Yeah, that's, again, a wonderful question. So, to answer that, I mean, so we're not the first program to fuse science and art, but I think where it differs from many I looked up on the web and I heard about is this interactive component between all participants, right? Where the transfer of knowledge happens manually by doing things, cognitively by exchanging ideas. And I mean, there are many programs out there where artists pair up with scientists and they take the science and interpret it or reinterpret it artistically with like new shapes, new forms, new colors, right? Where which is beautiful, but where I posed the question, how does the interactive or the interaction look like? What's the quality, and is there an equal share of opportunities in learned knowledge? And so that's the larger idea of what we pursue with our program. And I mean that generative moment, maybe not a moment, but a period, definitely the pandemic, where we witnessed how the new science was very quick to offer solutions through the vaccination, right? And then we just saw that some communities were very quick to pick this up, others hesitated, but then picked it up eventually, and others had a harder time, right? And you can look at that with it's a way we communicate, and maybe there is room to improve the way how we communicate in order to reach everybody better, right? And so
Everybody knows the vax.
Well,
The vaccination was developed at Penn. And I love that what you did in that moment, which is very important, thank you, was not, I mean, when I heard that people were doubting the vaccine, I just got upset. But I love that you thought, okay, this is what's happening. How do we address it? How can we make it better? Instead of just sort of being frustrated, you thought like, oh, well, they don't have confidence. We're not communicating well. How can we improve that? And I think that coming back to the theme of care, that's an incredible demonstration of caring for people and our society, and your project really demonstrates that, both of you. So thank you. Just think that's great.
Thank you. Yeah, and I mean it also, I think, reflects on a point that a way to change how we function as a society is to go back to very small communities, do the community work, and then hope that this trickles through a broader communities, right? And then institute change like that through that. Yeah, so that these are thoughts about that, right? And so um, as Marguerita highlighted during her slides, right, that it is a wonderful way to create community, where the other aspect is also that what Marguerita shared and many of the participants is that working with clay lets you forget everything else, right? And that caters to this idea of flow, where if you're able to blend out everything else and you just be concentrated on the matter in front of you, that this has a very high benefit in terms of mental health, etc. Right? Yeah. So to my shield. So I work over the past almost 10 years now on the question, and I listed a couple on the left-hand side, right? So there's this observation that many body functions, they vary over the course of 24 hours, right? If you do repeated measurements throughout the day, you will get different results because we change often each hour, right? And so then the question is, well, how abundant are these biological rhythms, right? Which functions are affected, etc. Another one is how do these change as we age, right? And there are so many age-dependent diseases like increase in blood pressure, etc., right? And do these biological rhythms have something to do with aging, right? And then the large question, how do these biological rhythms relate to health and disease, right? And as a scientist, of course, we set up an experiment, and this is shown on the right-hand side. I do studies in human participants, most often healthy volunteers, but also patients. And so here we just enrolled 10 young and 10 old participants. We measured many physiological functions through wearable devices and smartphone apps, but also collected lots of blood samples over the course of 2 days. And so on the next slide, this is just to give you a sense how variable we are over the course of 24 hours. This is blood pressure measured in young and old. Young are in green, old are in this orange. And you see below there's nighttime, morning, afternoon, and evening. And you see how the nighttime overall, the blood pressure measurements are lower than during the morning, especially afternoon, evening, right? But you also see that overall, the bars, the orange bars are always a little higher than the green bars. And that is exactly what we would expect, that in older folks, the blood pressure tends to be a little bit higher on average than in younger folks. So then on the next slide, we also see these rhythms over 24 hours in molecular features. And so here is just shown a gene that looks at gene expression. And then down you have the nomenclature. Yellow is morning, then evening, morning, afternoon, evening, night, and morning again, right? And if you look at these two curves, here the young are in blue, and in old, in red are the old. You see how in both age groups this particular gene oscillates over the 24 hours, right? In the next slide, And this is just a representation that we can look at all the gene expression, and the total is here almost 40,000 different gene transcripts, right? Each row is one gene. And here you see that the top third, right, there's a pattern shifting where on the left it's more bluish and on the right it's more yellowish, orangey, right? And that just highlights the difference in abundance of these genes, right? And that highlights how a third of all our genetic makeup oscillates over the course of 24 hours. And then, of course, on the next slide, we are not restricted to only look at genes, but we can also look at epigenetics, at proteins, at metabolites, and that's only half of it. And the green numbers, the percentages in cyclers, these just indicate how many features in each panel are oscillatory. And the big takeaway message here is that there are many, many genes, proteins, and metabolites which cycle over the course of 24 hours, right? And so on the next slide, so for the past almost 3 years, I'm looking day to day at blue and red for the most times. Blue the young, red are the old, right? And I look at data, lots of data. We counted all of our data, and to date, we collected 160 million data points in just 20 subjects, right? And that's a lot. And so how do you condense something like this onto a shield? And so this is the shield, how I conceived it, and Marguerita, in the conversation with her, was extremely helpful because I had 30 minutes where I froze up because I had a concept, but that was way too complicated to be translated into clay. And so I had then on the fly develop a new concept, and Marguerita was really instrumental to make that successful. And so here, just to read my blurb: rhythms govern our lives. The oscillations of many physiological processes and behaviors orchestrated over the course of 24-hour cycles rely on intricate time-specific molecular networks. To study this, we collect millions of data points from human volunteers. The particular challenge is to identify patterns, to understand how these patterns relate to each other, and to explore their association with health and disease. This shield symbolizes distinct data points coming together to form first letters, then a word with a tribute to rhythmic patterns. And so on the next slide, I provide the translation. So how to interpret an ocean of data. And please follow me counterclockwise. So top, red versus blue data points. These are the two experimental conditions, young and old, right? Then we start with and it's a word, the CNNH, and that highlights how existing knowledge, preliminary data, and hypotheses make low-hanging fruit easily discernible, right? So it's very easy to discern the C and the H there. Next up, the R. So here it gets a little more complicated. The top is still looking very much like an R, but the one edge is dots elevated with wire, right? So we have an increase in dimensionality here. And then the end of the R fans out. And so that is noise. And these are just all the factors like diet, like environmental exposures, which add noise to our data sets, right? Where we want to identify a signal. The higher the noise, the more difficult it is to see the signal, right? Moving on to the O. So before the kiln, it was an O, easily identifiable, but then the weight made the wire bend, and so it came apart, which adds this phenomenal aspect of that separation of data points challenges interpretation, right? So you look at data, right? And it takes you really a long time until you identify the meaning. And once you identified the meaning, it's like, oh, of course it's that, right? So once you know it's an O, you see it. Then the N, all of these are completely elevated by wire, right? More involved analytical approaches required to discover elevated data points, because so far we mostly stayed close to the surface. Then moving to the next O, and that's just a whole, and that is absence defines the meaning, right? So suddenly we need to invert our thinking to interpret that as an O. Then B, that spans almost all the shield, right? And that just highlights dispersion, how dispersion can conceal patterns in the data. Then moving on to the IO, that's very easy because these data are clustered and they are easily interpreted. Then the M, you barely see it on the side, very small signal. It has very clear shape, but the small signal jeopardizes the detection of that. And then the E signifies, well, if you expect to find all your data on the shield, you wouldn't find the E. And so then the signal outside of the predefined space jeopardizes detection in that case, right? And then the letter O in the chronobiome, right? We have it 3 times and it comes in 3 different shapes. So that's also often a pattern that the same signal comes in different shapes, right? And then lastly, with the luster, and that's hard to make out on the shield, but you see like little dots painted on the clay dots, right? And these dots of luster across data points, these connect actually into sinusoidal curves. So these up and downs, right, implicating the biological rhythms. And so that's the interpretation of that shield.
I think that deserves a round.
Moving on. Right, with that interpretation, it becomes very obvious, but you would be surprised how many did not see the word chronobiome spelled out on that shield, right?
Did anyone in the room see it? But
Okay.
now I do.
It's
Yeah,
indelible.
yeah. The next
Once you
slide,
see it.
please. And back to Marguerita.
Okay. So thank you. Gosh, Carsten, that was amazing. Opening our eyes. Thank you.
People I want the people on Zoom to know that the ceramic artists in the room were smiling, especially when noted that the kiln would change things.
Yeah. He used high temp kiln wire, but it was tall and those data points were the biggest ones. So the weight they bent over and they brought a new interpretation. So I had an extraordinary experience throughout both years of this residency, but especially a tender one when I got a phone call from such a dear loved family member that they were beginning a treatment called TMS, transcranial magnetic stimulation, to help with depression that was really resistant to meds and other things. So I was on my way into Penn on that day, and it was the first day of 2024 that these interns were presenting their research to us. So I get in the building, sit down, and then something told me to stay in that room. We broke into 3 rooms, and Dr. Deo was leading the group I stayed in. And Sneha got up and she presented on TMS. I had never heard of it before until the phone call 1 hour before. And there she was doing this research. And I thought, what a godsend that I can learn more about what my dear one is going through. So it also turned out the one research it went through both years of my residency is this one, TMS. First with Nick Balderston and Sneha, and in 2025 following Rebecca Voss's studies. So this, I learned so much, but I'm gonna this is me building the piece. Now the device that they place over the left part of the head, and when you get this treatment, it's putting some the energy waves into the part of the brain that governs, say, your mood. So when we're really depressed and shut down, it's very, say, the lights are off and it's not really, you're not really all there. So the penetration goes over like 36 days and it builds each time you go. So it gets stronger and it opens up and heals to brings more light in, so to speak, into your life. And so that's what these beads are. The device is, like I said, it's like an 8, a figure 8. And I saw it as the infinity symbol and feeling really focusing on the infinite healing that it may bring. So I built the infinity symbol that's floating over the shield, and the beads are the energy waves. And the next slide we can go to, and these are some really helpful illustrations. You can see the figure 8 device, and the energy goes through the handle, and when it enters each say the left coil, it goes counterclockwise, and when it enters the right coil, it goes clockwise. So then it's in pulses. So I decided to do the beads because I felt like it would be ceramically and structurally stronger to connect those instead of one long coil of clay. And I was really happy. I wanted to be really in integrity with the science and the way this device functioned, but it goes down into the brain at the most concentrated part, connecting point of the infinity, and that loops around into, keeps going. Okay, next slide. Thank you. Time. So next slide here, that is just soft bisque. I do an O10 bisque first, and then I can airbrush and handle it. I can still do some things that are just not possible to do in greenware, sanding and cleaning up. And then I fired the underglazes on. And then I airbrushed the clear glaze, which is the intense green, just in the area of the healing, and hand painted it. I went back in and came out. And next slide, we'll just see I'm painting the gold luster, the mother of pearl luster, incredibly carefully on the unglazed area, but these dots are just showing the healing. And it turned out that these beads, they also look like prayer beads to me after I made them, like a mala. And next slide. So here we did, the students this year presented at the ICA, and the Philadelphia Inquirer covered the story. It was just fantastic. They did such a good job presenting their slides. Next slide. And here's just some images of them presenting. Next slide. And so there, we'll go to the next slide, because Monday and Tuesday, it's been quite a week, but Penn hosted the International ITMAT Symposium in the 20th year, and we got to share the shields with this magnificent science poster that Carsten made. And we had the shields out and met with scientists around the world that came on Monday and Tuesday at Penn. Next slide. We're gonna go quickly here. Here's just some pictures of it, but it was really incredible to share this work, and we are preparing, next slide, for an exhibition. These shields will be installed in the composition of Metatron's cube. It's a sacred geometry. And you can go to the next slide, because this shows a detail. The lines are actually going to be the statements written by the scientists about their research and their healing, connecting all the shields. They will be installed. It'll be about 25 by 8 foot installation, and where it will be and when will be announced as soon as we know it.
Thank you. Thank you both. This has been really illuminating and exciting. We are a little bit over time, but just in case anyone has a question, maybe we can take one. Does anyone in the room have a question?
Maybe if you could go to the last slide, that
There's-
we have a QR code there for the audience to download. Yeah, on this one, to download the poster and the shields with the description for each shield.
Thank you.
Takes 10 to 20 seconds to load.
That's great. Thanks. I've known Margarita for several years, and every time we get to talk, I'm so inspired by the way your brain and your heart make connections with things. And I'm really honored that you came to share. And I'm so happy to meet again for the second time,
Thank
Carsten,
you.
and hear about your work. And it is this empathetic approach to science and medicine that we need in our world right now. And yeah, to bring it back again to care, it's care for ourselves and each other as humans in our world. So thank you so much for the work you do.
Thank you, Jen.
Thank you so much for providing this forum.
Thanks. Okay. Thanks, everybody. Thanks for coming. Have a great day. Bye. This will be recorded. You can share it with your friends probably next week. And come see our show, Clay is Care, and sign up for our symposium, which you can register for free through our website. Hi, Pierre. Okay. Bye, everybody.